Welcome to this first session of the one-month program on OmicsLogic Data Science. In today's session, our team will be sharing information, examples, and overview of OmicsLogic training resources designed for anyone to learn data science for biomedical data as seen in application to projects sourced from current topics in biomedical research. My name is Deepsha Biswas, and I am the OmicsLogic Community Manager responsible for daily interactions with thousands of users that are part of our bioinformatics community managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind OmicsLogic, share with you details about the upcoming OmicsLogic programs, and explain how the training resources we will discuss today will be used in the upcoming data science training program. OmicsLogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here includes bioinformaticians, computer scientists, and also our trainers, and some of our team will be presenting today. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. This program is designed to offer an introduction to statistical analysis and machine learning, and a broad overview of technologies and project examples to learn about and develop practical data science skills using user-friendly cloud technologies as well as R and Python coding. In this one-month program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the areas of bioinformatics and data-driven research. Let me introduce you to the program mentors and the speakers of today's session. Ilya Brodsky is our CEO and co-founder. His experience in research in academia has led to many research labs and university collaborations that has been recognized for the simplified approach to omics analysis for education. Dr. Harpreet Kaur is our OmicsLogic trainer and expert bioinformatician who will mentor sessions and guide you through the program. Her area of specialization is cancer genomics. Dr. Mohit Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. With this, I would like to pass over to Ilya Brodsky to continue this session. Thank you, everyone.
Thank you, Vipsa, and welcome back, everyone, to the first session of this data science program. So just to explain how these sessions are going to work for the remainder of the program, we're going to combine interactive sessions on Zoom, and in a little bit, you will see how we will try to make them interactive. We will actually ask you questions, and you will hopefully have the chance to reply and contribute to what we understand about our users. We'll also introduce some of the self-guided study materials that we'll be using throughout this program, as well as those that are available apart outside of this program. So we'll describe some of the resources that you'll be gaining access to. And we'll talk also about the platforms designed to make this experience seamless, because we see one of the major obstacles for people to start with data science is to be able to start quickly and focus on the topics and the projects that they're interested in, instead of doing the traditional hello world and then getting stuck with some of the next steps. So we are hoping to streamline, simplify, and accelerate your process into getting started with data science and getting started with bioinformatics. So I'll give you a little bit of a taste of that today, and hopefully throughout the program, you'll become more and more proficient using some of these tools. Okay, so one of the obvious questions that a lot of people ask is regarding the bioinformatics and the data science domains themselves. So what are the similarities and what are the differences between these two domains? Data science is a fairly new term that recently emerged and has become very popular. And bioinformatics is a term that has been around for a while now. And so our company specializes in bioinformatics. We provide a number of different resources as well as training programs for bioinformatics. And this is one of our programs, and this program is called Data Science for Biomedical Data. So what are the differences? Especially in biology and the medical science community, there is a lot of interest in data science. And so when people with background in life sciences with an interest in medical applications of data analysis come and ask, well, what's the difference between data science and what has always been around, which is bioinformatics, you tend to start questioning, well, what's the difference between these two and what are the specific details of each one? So when we talk about data science, it's important to understand that data science is the science of data. And one of the first requirements when you talk about data is to specify what specific domain is it going to be related to. So you do have to understand that each domain has a lot of experience and has a lot of knowledge that is required to be able to function in that domain efficiently. So that's why the specialization, that part, the pink part, is very important. And so in bioinformatics, that specialization is biology. In data science, that is fairly agnostic, so it could be anything. So really, the main difference between bioinformatics and data science is that specialization, the focus on biological data. It's important that when we think about what a bioinformatician does and what a data scientist does, they do very similar things. For example, you have to possess domain-specific data processing and quality check abilities. You have to have a general sense of how to do specific data transformations and filtering, understanding where the data is coming from. It's also important to understand that there are different types of informational data visualization and reporting tools that are going to be very domain specific. If we, for example, talk about specific types of visualization, and we'll give some examples and do those together, when we talk about general statistics or clinical reporting, for example, or specific types of information carried in specific types of applications of bioinformatics, right? So a lot of this also is inside a domain-specific range of APIs, databases, file types, etc. So all of these specific types of applications are very important to understand when we start talking about the differences and the similarities between bioinformatics and data science. Now, as all of you are joining from many different areas, many different countries, many different backgrounds, we have to find a common ground and an ability to be able to discuss the specifics of getting started with data science in this domain of biology and life sciences. And so that's why we're starting here from bioinformatics and data science. Now, as I mentioned, data science is the study of data. It involves developing methods of recording, storing, and analyzing data to effectively extract useful information. So the goal of data science is to gain insights and knowledge from any type of data, both structured and unstructured, which leaves us to the following question. What is the difference between structured and unstructured data? And then more specifically, we'll talk about what does that mean in the context of life sciences. So let's talk about the process of data and how that data evolves and changes forms as we go through this process. So the objective is to gain insight or be able to act on that insight based on data. So by analyzing data and understanding what's in the data, we can take action. That process goes from raw, unstructured data, which we can think about zeros and ones, so binary way of storing that data, all the way to then transforming that data into tables, structured data. Maybe those tables are annotated, they have some labels, so they help us understand in human terms what it is related to. Understanding relationships between different elements within those tables, within the structured data, we can map different types of relationships, and the connections between those different elements and between their annotation typically results in some kind of an insight. For example, what is important? What are some of the proportions? How do they relate each other? And what are some patterns that allow us to track that relationship? Ultimately, then we get to this ability to act upon what we understand, if it is understanding and containing that knowledge, or potentially developing a process. And that process helps automate and prepare the data for intuitive analysis and for insightful application of that knowledge. So the process goes from unstructured data. For example, we can talk about, in normal types of data, we can talk about media posts, emails, online reviews, videos and images, or speech and sounds. So really data that is contained in some kind of a digital format, but it is not easy to search and analyze. As we start working with that data, we start adding some labels and maybe meta information to that data. So that helps us organize things by folders. Maybe we create some kind of a data schema. Maybe we start organizing our emails in inboxes, etc. And then eventually we can prepare structured data. So that could be Google Sheets, or that could be Excel sheets, or that could be a database. And that database becomes accessible. That database becomes searchable. So it is ready for insights. So let's think about this together. Let me ask you the following questions to illustrate this process. So number one, what is your current level of education? So I know that many of you have already answered that in this chat, and we'll see how this is very difficult to manage. So people would say, I have a PhD, I'm a PhD scholar, I am from this country, and I specialize in this type of research. But to help us structure this data, let's provide you with a schema. So number one is you're in high school. Number two, you have an undergraduate degree. Number 3, you have some form of graduate degree. And number 4, you have a postgraduate degree. So please answer in the chat box. All right, excellent. So many of you have answered, and as you can see, looking in the chat box, you can see a stream of answers, and those answers range between numbers, as we assign to these different options, or some people would also add some kind of a description. So to illustrate the process, in a moment, we'll be looking at how to analyze such data and gain insights from this data. But before we do, let me ask you another question. And so the other question is, why did you expect from this program? Now, instead of answering this question in the exact same way, I will ask you to do something different, okay? And this will help us think about the structure of our data. Instead of answering just a number, okay? Make sure that for the second question, you are putting a number, for example, 2, dot, whatever the answer is. So for example, why do you expect, or sorry, the question should be, what do you expect from this program? So you can answer 2.1 is learn about omics data analysis. 2.2 is get familiarized with data science. 2.3, overview of to gain an overview of statistical and machine learning methods. 2.4, apply data science to a research project. And then some of you might be interested in multiple, so you will point 2.5 will be for multiple answers from this list. All right, so we have a lot of people here today, and so obviously there will be a lot of different answers. Okay, and thank you for providing this answer. Let's now think about how the answers themselves can become information, right? This is raw data. Anyone looking at this can spot numbers coming in on the screen. You can see the variety of answers that are coming in and the variety of formats, okay? But now we will try to gain insight. What does this information actually carry? Okay, so the way that the unstructured data comes in is in the following way, right? So some people start answering early on, some people are putting in different types of answers at different times in different formats, and then we can process this unstructured data and convert this into structured data, which allows us to very simply translate the information from that unstructured data into some summary. For example, many will answer that they would like to apply data science to a research project. Okay, or for example, we can look at all of the participants and we can see that many are postgraduate, right? And so as we continue to ask these types of questions and provide the users with a schema, with an organization of that data so that it is easy to process, it is very easy for us to translate this information into an actionable type of a resource. So that's really the value of the understanding of data structures and preparing a format that is specific to the type of interaction we have. Now, this is a simple example. This is an example that allows us to learn about you, the participants of this webinar, but also for you to start thinking about the significance of how the data is organized. So let me ask you another question. What is an example of unstructured data in research? Okay, now you don't have to follow any schema. This is more of a discussion. So what do you think is a way, is an example, sorry, of unstructured data in research? Okay, so someone is answering documents or raw data. Let's be more specific. So give me some example, okay, non-analyzed reads, okay, so maybe sequencing reads, results from experiments, so direct results from experiments, raw genomic data, excellent. Images of specimens, okay, very good example. Observational, okay, Miley is answering. Soil samples from garden, okay, so soil samples if they're stored somewhere, that's interesting. Raw sequencing, Sana is answering. Jefflin, sorry, is saying video, okay, so maybe some recording of an experiment, okay. Clinical notes, a very good example. So this maybe is related to clinical research, okay. So literature, any numbers generated through experiments, so raw PCR data or imaging picture data, okay, excellent. Okay, let me ask you another question. What is an example of actionable data in clinical practice? Okay, so if you think about clinical practice, okay, a clinical practice is when an interaction happens between a clinician and a patient, or some member of the clinical community and the patient. Can you give me an example of actionable data in clinical practice? Okay, so people are saying EHRs or MRI data, clinical application forms, okay, blood pressure, X-ray data. Okay, so excellent examples. Let's think about what makes them actionable, right? So here we were talking about unstructured data. For example, we talked about raw sequencing reads, right? And then how do we translate that into an actionable data? Maybe a report like suggested by Anjali, or a biomarker suggested by Anuradha, right? So we have a biomarker, an indicator that is known to be associated with some clinical condition, right? So that's really what makes it actionable, is a studied and characterized association. Okay, charts and graphs. Okay, so maybe those are some insights. Charts and graphs are some insights. And then what makes them actionable if those insights provide an established way to talk about some clinical and studied condition, right? So it's the association. What we're trying to say here is that we have raw data and then we have an association. Okay, let me ask you another final question. What is an example of a data process? Okay, so think about those two things that we talked about, number one and number two. We have raw data. You have a lot of examples there in the chat. And then you have some actions that could be made based on analysis of that data. So give me some examples of a data process. Okay, so filling data, genome assembly, charts and graphs, so making charts and graphs, multiple sequence alignment, okay? Replication, okay? So let's think about what other processing we could think of in the context of this life science data. Okay, excellent. So Genova is saying differential expression analysis, or QSAR, okay? So variant calling, chromatogram file trimming, microarray. Okay, so again, what's important here is to remember that there is a difference between, for example, microarray. Microarray could be the type of data, and a process. And so variant calling is a great example, right? Because you have a lot of raw data, you have a lot of aligned reads, and then you're actually calling, you're looking for specific types of variants. So we'll talk about that in a second. But as you can see from this kind of a thought process, really the objective of this data science in biological data, in life sciences, especially in the domain of this biomedical research, is to be able to incorporate the data science principles into the processing, organization, analysis, and insights that come from associations between clinical and omics data, between the raw data and the structured data to provide us with insights. And the objective, a lot of times, is to really provide insights about diagnostics and about selection or development and design of therapeutic interventions. So that's what our program is going to be focused on. And that's how the OmicsLogic data science program is going to be different from some of your other data science resources and programs out there. Instead of just talking about general data, which we could be talking about surveys, which is what we did today, or we could be talking about analysis of sentiment, why and how do people feel about different things. In this program, we're going to be talking about omics data. And specifically, we'll be focused on the association, the relationships between clinical phenotypes and their utility in different types of examples. And on the other side, the unstructured data that comes out from different types of techniques to generate data in life sciences. And that will include particularly the omics type of information. So omics, we'll talk about that in a second, what type of data that is and what kinds of insights that could provide us with. Now, for all of you that have submitted your answers about your background, about your location, maybe even about your current institution, we ask all of you to take a look at the Introduction to Bioinformatics course. This course is not only a good overview that could be very useful for those of you that are interested in bioinformatics, but also it includes a survey. And that survey is going to structure this information for us to be able to analyze and understand the interest in the community in bioinformatics, and also provide you with some applications of bioinformatics in different domains. So all of you, if you could please look in the chat, there's the link to this course, Introduction to Bioinformatics. It's a free course, so anyone could take this. And in the first lesson of that course, you have the survey, and that survey will provide us with this important information because we are constantly developing ways and tools to make bioinformatics more accessible, and that is actually our company mission. And the way we think about bioinformatics is that from becoming a very niche area of life sciences, very unique and specific domain in life sciences, it is actually becoming an important part of skill sets that are needed across the whole range of jobs and positions within the life science community, from people that actually generate data in the labs, okay, so lab technicians and people that deal with samples. Their objective as we move into the future of life sciences is to generate more and better data. They have the ability to control quality. They have the ability to control the annotation and the details that they provide with the data that's coming out from those labs. And so it's very important to be able to understand the use cases of bioinformatics and the different skills and be able to be proficient with analysis and handling of that data. Obviously, the people that deal with data have to understand how that data is generated and how to use it at the other side of it. So they're really the link between the people that generate the data and the people that use this data. And many times, what the normal way that you kind of look at bioinformatics from a perspective of just an analyst is specializing in the processing and the insights part of it. But I think that as bioinformatics continues to expand, it becomes more and more practical. And so people have to understand how data is generated and how it is used in the context of making clinical decisions. And so that's the objective, is really to provide bioinformatics training to find a way to look at biology as a data science and to understand how it helps us provide, how it helps us make those links between the way the data is generated, the way it is analyzed, and the way it is applied. Now, for any one of you that is joining us for the first time, I also want to request that you not only look at the Introduction to Bioinformatics course, but you also start building up your profile. Okay, again, this is something that you can start doing for free, but it's an important part of letting the world know who you are and why you're doing what you're doing. And I think it also can explain how important it is for those of you that are going to continue your journey in bioinformatics to provide the information about yourself in an organized way. And I'll talk a little bit more about how you can then build up your profile and advance your position within this world of bioinformatics. So first of all, there are a lot of tools to make your bioinformatics profile more stand out. So make it more accessible, more visible, more searchable, okay? What's an important part is to name who you are, so give it a full name. It's important to put up a face, a picture, an avatar, whatever it is, and then to start developing that profile. So really to make mention of your publications, to make mention of your social media profiles, to join different groups by interests, and to start showcasing your skills and your projects. And so this program will actually help you make sure that your profile becomes more searchable, more visible, and stand out from the rest. And that's one of the objectives that we have here, is to help those that are starting with bioinformatics, perhaps coming from other life science domains, generate a profile that stands out and that is impactful. Okay, so the way we will do this is we will track your activity throughout this program. So as you're joining this program, create your profile, you start filling out different surveys, start completing different courses. That process is going to help you really showcase what skills you're gaining and what topics you are covering in the process. And so here are some good examples from people that have recently joined a program and are starting to make some progress. So you can see, for example, April Zhang here filled out all of her social media profiles, and she's joined the OmicsLogic biomedical data. You can see Marek, for example, or you can see Hassan here. Every person has the ability to really build up their profile. And so what is the ultimate outcome from this process of building up your profile? The final outcome is that after completing this full program, you are going to build up a profile that stands out. How will that happen? So for example, here you can see Mikhail. He joined the program. He has completed multiple programs and now has certificates on his profile. And he is also one of the top profiles on our leaderboard. So you can see that our company runs a leaderboard of people that complete courses and do different activities throughout the program. And as a result, you are more easily found within this community. Now, also, for those of you that are doing this, you will be able to provide some feedback on the program, and we'll actually highlight your project and your responses to the different questions. And I think it's very important for us to build up this community, to learn from each other, learn how do you leverage these resources and what you can do with them. And so many of the participants from our previous programs have gone on to become professionals in data science and in bioinformatics. And so I hope that for all of you joining us here for the first time, this is the journey that you're embarking on with us together and with our whole community. And to do that effectively, we will try to make all the efforts on our side to help you get to that level. So again, I see that some people are requesting those links again. Thank you, Vipsa, for posting that. Okay, so let's talk about omics data, and let's talk about what are some of the challenges and opportunities with big data and why data science is even needed, especially in the domain of biomedical research. So as you know, there is an exponential growth in the resources that are being made available on public repositories. And what that means is that we are sitting on this valuable resource, which is unanalyzed data. So even though you can see here the number of GenBank sequences and the number of users is starting to overtake the number of published NCBI resources, but also you can see that in the end, a lot of the data, the raw data that is being posted on these repositories is actually not completely analyzed and annotated. And I think it's both a challenge for us as a community to gain insights from this unanalyzed data, but also it's an opportunity because as these are unreported insights from data that already exists on public domain repositories, we can contribute and we can gain insights, develop novel hypotheses, and also be able to handle this data in a very efficient way to enrich and empower our research. Now, one of the challenges with big data is that this data has properties that require efficient tools to process, analyze, gain insights, and structure the data efficiently because this data is constantly changing. It comes with a variety of formats, and it has very specific domain questions and technical variation that we need to know how to deal with. So, for example, let's take the example of next generation sequencing. How many of you here are familiar with next generation sequencing? Okay, if you are familiar with next generation sequencing, what it is, please put in 1. If you are unfamiliar with what it is, please put in 2. Okay, so number 1, if you are familiar. Number 2, if you are unfamiliar. Okay, excellent. So I see that many of you here are familiar and some of you are unfamiliar with it. So let's briefly, before we go on, explain what next generation sequencing can allow us to do and what is this data, this omics data that could be genomics, transcriptomics, epigenomics, etc. What can it give us? Okay. Okay, so next generation sequencing or omics data, as an example, let's take next generation sequencing data. Once we characterize the samples that we have pictures, for example, here of cells, and these pictures can help us characterize what these cells look like, what is their shape, how are they different from other types of cells, right? We can really categorize that data. Now, we can do this in a simple way, looking at the picture and kind of describing it in words, or we can process these images, analyze them computationally, and generate a lot of that data, which together will be called phenomics. Okay, so this is the study of the way things are looking and characterized. As we look beneath the surface, besides what it looks like on the surface and how it functions perhaps, we are going to discover molecular signatures of what is the cause to lead to this phenotype. And so that type of data can include genomics. Okay, in terms of genomics, next generation sequencing genomic data can provide us insights about variants, how we can compare DNA sequences of these cells to other normal cells, and what are the differences, what are the variants in the DNA code. We can also look at this emerging field of epigenomics. And one of the questions that epigenomics asks is how does this process of going from DNA variants or non-DNA variants can cause differences in gene expression. So when we talk about the phenotypes are produced by proteins that are functional, and the cell itself is regulated by multiple processes. And not all of these processes are genetic. They're not resulting from changes in the DNA. And so as a result, we have this field of epigenomics. It has become a prominent part of next generation sequencing because we can study the role of microRNAs, non-coding RNAs, and all kinds of variation that leads to gene expression changes but does not come from DNA. Now, ultimately, we then come to the expression of RNA. And the expression of RNA could be mRNA or non-mRNA, but transcriptomics is the field that focuses on the analysis of mRNA. So a lot of times we think of mRNA as really the immediate step before protein translation into protein. And so it's a good proxy, a good measurement of what happens at the protein level. So next generation sequencing is a technology and has specific types of machines, but those machines can produce multiple types of insights. Same data, different types of insights. So if we have multiple types of data one type of data for multiple types of insights, the question is, how can we find those insights within next generation sequencing data? So the answer is, we have to process this data. And the specific types of processing, the specific types of analysis that translate this raw sequencing data into tables is the process of what we call bioinformatics pipelines. So we have to take this raw sequencing data and go through multiple steps to be able to translate this data into a particular structure. Okay, let's take a look at how to do this together. Bipse, if you could please post the demo account information so people can log into the platform, we will illustrate how to do this. So I'm just going to pause as I go to the next slide, and please go to this link, server.t-bio.info, and we will take a look at how processing is done to generate information that could be used for analysis of this data. Okay, so I will go to server.t-bio.info, and you have here in the chat your details so you can log in. Okay, so once you log in, please let me know. Okay, so once you're logged in, you should be able to see what I am seeing, which is the T-Bioinfo areas of analysis. When you log in, please put number 1 in the chat box. Okay, for those of you getting here for the first time, you will see here login, and you'll be able to log in. Okay, great. So Apurv and Dr. Hundama, Mustafa, great. So it's great to see that you're logged in, and you can start exploring and kind of seeing what's available as we wait for others to log in. Once you're logged in, you should also see right here, I see my account, but you'll be seeing info at Pine Biotech. So you'll be seeing here that you are logged in. Okay, did anyone else manage to log in? All right, great. So let me explain what this is. This is the T-Bioinfo platform, which was designed by the Tabor Bioinformatics Research Center to simplify processing and analysis of large data sets like next generation sequencing, mass spectroscopy, structural data, and perform multiple types of integrations, analysis, statistical analysis, annotation, and also train machine learning models. So we'll talk about all of this in the context of this data science program. Okay, for those of you that cannot log in, Bipsa, maybe you could provide another, okay, thank you. So there's an alternative account. Too many people logging in at the same time is not going to work very well. So we've provided multiple accounts. So if you did not log in with the first account, try the second account, and you'll be able to see the same thing. All right, so for those of you that have logged in, you'll notice here, we'll take a look today at RNA-seq data, so transcriptomic data. So please look at this first section and don't click on it just yet. Click on this down arrow. So once it clicks, you will see there's a few demo options, and we will select the first one. So demo RNA-seq. Okay, so once you're in here, you'll see that there are a number of buttons, and these buttons each represents an algorithm. This algorithm will take the source data and do one function. Okay, so typically, for example, the start button will load in all of your data. So let's click on the start. In the start, you will also learn about the next steps that need to happen, including the processing or the pre-processing of data, the raw reads, and we'll talk about this in our next session, actually in much more detail. But first step is to do pre-processing to make sure that the reads are clean. Then we do mapping only on the good reads. We will do mapping on the reference genome. And then we will quantify expression levels. So we will look at the level of expression of genes, okay? And here's an example pipeline. You can actually select Trim-O-Matic, PCR clean, then go to Bowtie2T, RSEM expression table, and then end. Okay, so I'll give you a couple of minutes to prepare this pipeline on your own. If you were able to create this pipeline on your own, please put in number 2. So I know that you have completed this pipeline. For the rest of you, if you're not able to do this or you're not logged in, please review the login information above in the chat, and we're going to try to do this together. Okay, Glory, thank you. He's completed. Anyone else? Okay, and for those of you, okay, all right, excellent. If you're just logging in right now, you go to the areas of analysis, okay, and here under transcriptomics, you click on the down arrow, and we're selecting this first option, RNA-seq cell line project. Okay, so for those of you that are logged in, how to log in. So for those of you that are not logged in, there's a password and an email address that you can use that was posted in the chat. Okay, and then you can gain access to this demo account. Okay, so for those of you that got to the place where I am at, okay, so you go to start. You see here an overview of 3 major steps to process next generation sequencing transcriptomic data. And now you have to create this pipeline. Okay, so try to do it on your own. After you've tried to do it on your own and were able to create this, put number 2 into the chat box. If you have not been able to do it, then just give it a couple minutes and we'll do it together. Okay, and thank you. Mustafa is helping out here as well. For those of you that need to log in, you will have to, okay, great. So Dr. Nduma is in, excellent. All right, so let's do this pipeline together. So here we have the button start. Once I click on it, I need to click on OK, adding it to my pipeline. Now you can see that it is blue. The next step is to do pre-processing. So we'll use Trim-O-Matic, which just simply trims the reads from bad quality and technical sequences. PCR clean, which deals with PCR bridge amplification. We'll talk a lot more in detail about this in our next session. Then we'll do mapping onto the reference genome. You can use HiSat2. If you want to go back, you right-click, and you can also go to Bowtie2T. So remember from the example that we saw in the start button, it was actually Bowtie2T. So Bowtie2T is an algorithm to apply mapping on the transcriptome, so just the expressed genes. Okay, so now we did our pre-processing steps, our mapping, and now we create the RSEM expression table. And what the RSEM expression table will do is actually prepare a table of expression based on how many reads were mapped to each element, genomic element on the DNA, which includes genes, transcripts, exons, etc. So now you will have a gene expression table. So we are done with our pipeline, right? So we've taken raw sequencing reads, we processed them, we now map them onto the reference transcriptome, created a gene expression table, click on end, and OK. And now you will see that there are some files that we can work with, and now we can do downstream analysis, right? So now we can look for trends, we can look for patterns, we can look for associations only after we prepare and process this data. So let's think about this process that allows us to transform raw sequencing data into specific types of structured data that we will be working with throughout this program. Now, it's important that in every lesson that you will find, in every course that you will find, there are going to be practical assignments using this T-Bioinfo platform. This platform can allow you to study different types of omics data, including genomic data, transcriptomic data, metagenomic data, and examples from all of these types of sequencing data will be used in the data science program as we continue to work throughout the program. So some of the things that we'll be doing is processing. A lot of it will be downstream analysis. So we will look at different types of variations and we'll try to analyze these variations for insights into biology, into the meaning of what the data describes, okay? So as you go through this process, remember that this process is actually describing with color-coded maps what is the logic behind this analysis, okay? So for those of you who are thinking about the program, we are going to cover each one of these steps and describe how these steps form a logical approach to the analysis of next generation sequencing data. The colors help you remember what are those steps and how those steps are logically connected, okay? As you go through, each method will be explained. So there's going to be a link to the article. There's going to be an explanation behind the principle of why we're doing what we're doing. And if you would like to make this a part of your own analysis pipeline, you'll have a link to where you can download the script and actually run it on your computer, on your server, or use it on this platform. Now, all of these different types of methods produce a variety of different outputs and inputs for whatever next step in your analysis is going to be. So what we're going to do is we're going to discuss the different formats. We'll talk about data files and data formats that go into each one of these methods and the different data types and outputs that are coming out from each one of these steps. As you practice and do a number of these different exercises, you will see that this guided process is going to prepare you to be more independent and to be more capable at doing what you're doing so that you can now apply some of these methods to the data that you find on NCBI or maybe you generate in your own lab. And it's important that we will also be talking about reproducible workflows. So these reproducible workflows are going to allow us to share information whenever you complete your analysis with someone else that will ask you, well, how did you actually generate this data? Okay, so at this moment, I'm going to take a pause and ask if there are any questions about what we did so far. Are there any questions about what we're doing? Okay, so Aiswaria is asking, does the knowledge about how the raw data is converted to structured one through this pipeline is shared in this course? Yes, so that is exactly the objective, is to explain each step, first of all, at a very high conceptual level so that you understand how the logic puts them together, and then going deeper and deeper into each one of these methods to understand the variation, to understand the settings, to understand the process, and also understand the objective. What can you actually do with this type of data? Okay, excellent question. Will it be useful for someone who is not a biology student and someone working in software development? So if you're working in software development, it's important to understand that this type of data is growing exponentially. And as a result, there's a lot of interest in finding people that understand data sciences and know how to deal with data effectively to help analyze and extract insights from these types of data sets. Now, that is your traditional bioinformatician. and bioinformatician jobs are available. There's many bioinformatics companies, and there are many companies that are pharmaceutical companies and biotechnology companies that are going to be interested in finding someone that can deal with this data efficiently. Okay, can we learn CAD here in this course? So the course is going to be focused on data science. So it's going to be focused on how to take process data and find patterns, analyze those patterns using statistics and machine learning, and to be able to design a data science analysis process that is reproducible and effective. Can we execute our real-time projects using our platforms by paying apart from this training? Yes, so definitely this is a training program for those that are interested to gain skills and those that are interested to complete a course. The platform itself is a research platform, and Beepsa, you're welcome to put the link to the pricing and the details about the different research licenses also for those interested. Do we have any class for a chemistry student who wants to jump to bioinformatics for a PhD course? I think this introduction is going to be a useful start. Okay, so for those of you that want to just kind of explore before you jump into this, I highly recommend taking some of these courses that go from very beginner level courses to more and more advanced. So you can see here, Introduction to Bioinformatics, Genomics, Genomics 1, 2, Transcriptomics 1, 2, 3, and 4, etc. So you'll see more and more advanced courses as you join this program. I think it's important to get an overview of a variety of different types of omics data, understand what's included in different stages, and then eventually to develop a project. So the project is going to really help you demonstrate what it is that you want to do, but also understand how it's all working for the specific types of applications that you're interested in. Okay, currently we do not offer a course on proteomics or metabolomics, even though there is a section on mass spectroscopy data. If you're already able to use this in, if you understand what the logic is, you can use the sections here for mass spec data. All right, thank you very much for your questions. And we will continue to talk about these types of applications in our next session, which is coming up very soon. But let me go back to the presentation to kind of pick up on where we are after we analyze this data. What are we going to do with the analyzed data, right? So once we analyze this raw data, we have our table of expression, or we have characteristics of a protein, maybe mutations that we know of their specific position and function. We're going to be able to start asking questions, okay? Remember how we took the data from the chat and translated that into some insights and understood proportions between the number of participants that have a PhD, that have a high school degree, or have an undergraduate degree? Those are insights. When we talk about insights from big omics data, from these huge data sets, we're talking about the ability to find relationships that can be actionable, that you can do something about. Okay, so in the context of biology, that means if I understand that here we can see mutations on the spike glycoprotein of COVID-19 and the ACE2 receptor, I can think about intervening in the connection between the two and maybe blocking the virus from entering a cell. If I understand how there are biomarkers that can explain differences between different types of cancer subtypes, I can ask the question, what can I do about those genes that are overexpressed in a particular type of a cancer? If I understand that there are specific sequences that are found, and those sequences annotate or mention for me that this is the beginning of a gene, I can find maybe a microRNA that will bind to that region and block expression of a gene. For those of you interested in structural biology, I can understand the physicochemical properties of molecules, identifying targets for the different molecules on a protein structure, and try docking them so that I know that they fit well, so that their chemical properties interact with the chemical properties on the protein, and that interaction is strong enough for me to understand that they will function and activate or deactivate some portion of that enzyme or protein. Okay, so really the idea here is to learn about complex relationships that are becoming very, very important as we study biology at the subcellular level. Now, to understand all of this, it's not enough to give you some general information about all of these topics. What we'll do is we'll take a number of projects. These are going to be sourced from high impact journals, peer-reviewed journals, with data sets coming from those journals directly and analyzed to provide us insights into diagnostics and cancer biology, single cell and tissue composition, and microbiome and how it relates to infection. Okay, and yes, Shayeri does, the full program will include a certificate from Pine Biotech, but also from the Tauber Bioinformatics Research Center, which is an academic institution. Okay, so as we think about these projects, you will just get an illustration of how the different steps and aspects of data science fit in to help you produce meaningful results. So here are some of the skills that you will learn about and you will be able to apply at the end of this program. You will see the relationship between experimental design and data collection. As a result of understanding how the experiment was conducted, you will see the need for pre-processing and cleaning so that you can organize and prepare the data for analysis, eliminating technical variation, addressing batch effects, and any kind of technical specification of the machine and the lab where the data came from. After that, we will talk about exploratory data analysis. We'll talk about how to see what types of patterns and general statistical properties can help us understand the data we're dealing with and generate a hypothesis about the different types of comparisons or maybe specific types of data that we have available for training. We'll talk about data mining. So oftentimes we can visualize our data and we can also look for patterns, but there's so many patterns to look for, we really have to mine the data for something informative and interesting. So we'll talk about different tools and methods to do that and how to deal with the data sets based on whatever our next objective is. Now, the process of data analysis will include some statistics and machine learning. So we'll talk about the difference between traditional statistical analysis and what is called machine learning or deep learning, when we actually train a model and that model can help predict or help us understand what's inside the data without necessarily understanding exactly what this means from a biological or a clinical perspective. So we'll talk about some of those important things, and we'll talk about how to go from a trained model or a statistical analysis to inference. Now, when we do that, we'll cover a number of different methods, and those include different methods of data mining and machine learning that are becoming commonly used in a variety of different biomedical research papers, bioinformatics research papers, biology research papers, and even are used at the more clinical research level. So we'll talk about things like decision trees and random forest, linear discriminatory analysis, and stepwise LBA. We'll talk about hierarchical clustering and k-means clustering. And we'll talk about the differences and similarities between these different types of methods and how they can be used to gain insights from big data. Once we do that, we'll also talk about the process of prediction or inference that is used in the context of statistical analysis. We'll talk about the different data types, and we'll talk about how those different data types should be handled for us to be confident and for us to know that the prediction is valid and for us to be able to explain what this prediction means. We'll also talk about some basics about data science, where we think about the different types of data and the appropriate methods of visualizing that data so that you can present your analysis at the end and be capable at explaining and visualizing data effectively, knowing specific types of approaches that come from an understanding of syntax behind R and Python. So we'll cover a little bit of both of those languages and be able to talk about how to prepare and work with these different types of data. Okay, so I think it's very clear that when we talk about data from chat boxes or emails or maybe even journal articles, it's fairly simple. But when we talk about omics data, all of these different steps are maybe not always clear-cut and understood by everyone. So let's take an example. Here's an example of genomic data or genetic data from a clinical panel designed to inform a clinician about risk for mutations in breast cancer. When we talk about mutations in breast cancer, probably everybody is familiar with the BRCA1 mutation, right? So BRCA1 mutations have specific weight that they carry from a clinical perspective. When we want to test for those clinical indicators using next generation sequencing, the first step is to generate that data. So you generate that data, and that is done in the wet lab, right? So from before the wet lab, maybe a clinician assigns a patient to this lab, and then the data is generated. After that, the process of analyzing that data will look at the short reads that come from a particular gene, the BRCA1 gene. The BRCA1 gene will be compared between an individual and a reference, and then the variants that are most prominent in that sample will be reported. And oftentimes that includes being able to interpret what is found, because you will oftentimes have a number of specific rules and guidelines to look at the source data, look at the analyzed data, and prepare a report. So that is one of the things that we will do in this program. We'll actually go through this process of looking at the raw data, looking at the structured data, looking at those guidelines, and trying to piece all of that together. When we do that, we will rely on algorithms, okay? So the important part of this data science program is to not only understand conceptually, but to be able to work with algorithms, tools that people have designed to actually extract meaningful insights relying on a computer. And so here what we'll do is we will take, this is just an example, we'll take a look at how do we take all of the available features, whether they are variants or whether they are genes and their expression data, and work with algorithms that are called greedy algorithms that help us do feature selection. So the process will be by iterative, looking at all of the features, using statistical approach to understand the value that this specific feature carries in the overall data set, and then try to maximize the separation between classes so that we can go through the process of feature selection and improve accuracy of the prediction of our model. And so we are going to look at all of these different steps, both from a conceptual and a technical perspective, so that after completing this program, you yourself will be proficient at reading, interpreting, and evaluating the relationship between algorithms, data, and insights. In the end, you will be able to place yourself in a position of a clinician that is dealing with taking samples from a biopsy, a tumor biopsy, separating that data into data that could be translated into DNA variant calling, okay? And then compared to your database of big data, let's say on the Cancer Genome Atlas, you have thousands of patients that have their tumors characterized by different clinical phenotype. and use the analytical approaches that we are discussing here today to gain insights. Not only that, you will also be able to report your insights in a meaningful way to your partners that you are dealing and collaborating with so that they understand what is in the data without having to ask you, what exactly did you do? So we'll learn about some of the commonly used approaches for bioinformatics analysis, interpretation, and visualization that will be commonly accepted by people on both sides of the aisle, so to say, right? So both the technical people that deal with data and the people that interpret the data, that understand where the data came from. Okay, so for any other questions about the types of project examples and resources that we will be using, I recommend looking at the edu.tbioinfo platform. That's a repository of courses and projects, and you will be able to see here a variety of different resources, all being included in this program, that include epigenomic data, metagenomic data, genomic, and transcriptomic, as well as some of the courses on BioML, or machine learning for biomedical data, as well as some introductory bioinformatics courses. I'm not going to go today through all of these different resources, but you will see on the program, which is a place where you can find all of these repositories, all of these different resources. Once you complete your registration and you're a part of this program, you'll be able to see how they are all organized right here under courses. So if you go under courses, you'll be able to find what courses are included, what projects are included, and how this program will help us go through all of these different steps to learn about these different skills. So today, we're just starting out with Omics and Introduction to Data Types and Properties. As you continue, we will start talking about in detail some of the specific steps of processing this data. Then we will go on to talk about coding, how to load and process the data, as well as filter out unwanted variation and understand an overview of what's in the data, and continue on to then speak about exploratory data analysis and visualization. Then we will talk about how to actually design a full script that goes from one step to the next, and what are some of the outputs that you will expect to find. And then we will talk about machine learning. So we'll dedicate a whole session on to machine learning and artificial intelligence, which of course are very important today in biomedical research. We'll talk about statistical analysis and how to do biological interpretation of identified features and variation. And then we will talk about supervised and unsupervised machine learning, as well as production, how to actually do validation of your model, and how to make sure that the type of insights you're producing are valid and reproducible. So we'll talk about all of these different parts of it. And so this is kind of a brief overview of what you will expect to see. Now let's turn to the final area that I think is going to be very important for all of you that are joining this platform. So as you probably know, data science for biologists is a pretty big jump, right? So if I'm a biology student or I'm a clinician, I am actually going to be interested in all of these topics, but I might have doubts. Am I actually going to be able to jump in and do this myself, right? What are the technical requirements that are needed? If you are an engineering student, you're interested in data science, you probably know that many people that speak about data science are going to be talking about different coding languages, okay? So this is a survey from Indeed.com that was done in July 2018, so about a couple of years ago. And you can see here that one of the prominent languages in data science is going to be Python. The second one by rating is R. And so there are two things here, right? So one, for people that don't know any coding, how do I even get started, right? For people that know coding, it's what language should I be proficient in? This program is not going to be about programming. This program is going to be about data science. So with the outbreak of data science revolution, there's now a war between R enthusiasts and Python fanatics. and people start to compare and contrast them 1,000 times with detailed listings of their respective advantages and weaknesses. And so this warfare actually leads to this misconception that maybe as a data scientist or someone that wants to do data science, you should always focus on either one or the other. And this is actually bad advice. So the key to becoming a data science professional or to learn how to use data science for your needs is not to focus on one particular method of doing things, but it's actually to focus on data science, being able to work with data, being able to ask questions of data, and being able to work using data together with other people, so to make it reproducible, visual, and insightful. So that's why here we will not focus on the specifics of the different languages, and this is, by the way, in 2019, but actually to be able to use it as a biologist, as a technician, as a person who's just interested, to be able to get started. So let me show you how we're going to achieve this, okay? And this is, again, not going to be for anyone that is just interested in data science, but we're going to be focused on data science for biological data. So please come with me to Omics, to code OmicsLogic.com. Okay, this is the site where you can also, thank you, Beepsa, this is our coding playground. This is where anyone can get started very quickly and learn how to program enough to be dangerous in biology and life sciences. Okay, so please let me know once you're here. This is the main site that you would go to. Okay, this is free registration. Again, let me show you the registration itself. You just put in here an email address and a password, okay? And once you do that, you will be asked to put in your credit card details. Again, this is not going to be used up until you start getting into more advanced courses. And if you join the program, you'll actually receive a discount code that you'll be able to do for free. Okay, did anyone, let me know once you're here on this link. Abdel Jabbar, okay, Glory, thank you. Abdel Jabbar, if you have registered, again, this is a free registration. You should be able to see what I'm seeing now. Okay, and I'll just, please let me know once you're here, and I'll just quickly review the questions here posted. How this data science course is different from other data science courses for a non-biology student. Again, once we have the structured data, the model building going to be the same with data from other sections like financial data. More or less, you'll use a lot of similar approaches, but you need to understand both where the data came from, so what does it describe, and what are the insights mean, right? So that's the challenge specifically with biological data. It's not the actual process, it's understanding where the data came from and how do you actually report and analyze the data to make it biologically meaningful or actionable for clinical insights. Okay, is that the same algorithm used in other data sciences also used for biologists? More or less, the actual analytical or machine learning approaches, they will be very similar, but there's a lot of variation just based on the type of data. Okay. Okay, did anyone, was anyone successful at getting to OmicsLogic.com/courses? Okay, let me know once you're here. We'll actually do an exercise together. All right, excellent. For those of you that are here, let's go on to Introduction to Data Science in R. Okay, so once you're in this course, let's go to how to load data and check for variable types. Now, these courses are going to go from very basic to more and more advanced as you go through the program. So first in R, you can import directly from the code section. simply by writing an import code. So again, this is a function that tells our program to load in some files stored on your computer or stored somewhere on the web and tell us what's inside. Okay, so let's do that together. Here we have a number of possible data types that we will be importing. For example, we can take a TXT file, or we can take an Excel file, or we can take a CSV file. And you can see that we are creating an object, a data frame, and we are inputting that file. Now, based on what's in the file, we need to specify sometimes if there's a header and maybe what type of a separation there is. Okay, so this is a very simple procedure intended to get you started. Okay, and even before you start setting things up on your computer, you can do it right here inside these little exercises. So for example, here's a link to a TXT file. Okay, so let's do that together. You can see this is the code block. It's editable. Okay, I can say here anything I want. Okay, when I use the hashtag, this is the way to comment out what I'm writing. And what you need to do here, you can see that the code is just to read a table, but this is a path on my computer. So instead of this path on my computer, what I need to do is I need to copy this link, which is a file that is not on my computer, replace this portion right here with that link. Okay, and then click on run. Okay, let's see, probably I did something wrong. Okay, so there you go. All right, so we ran this code and we have here flower, different types of flowers, and the tree and the bird, right? So we have some biological data here that we've analyzed, or we've loaded some biological data. We can look, of course, at what this data is. Okay, so let's take a look. This is what the data set looks like, and this is what indeed this little program loaded for me. Okay, so try and go through the rest of them. You'll see that there are multiple types of data. You can specify different things, and you can also try with a number of different data sets here below. So each lesson that we will do, you will actually going to go into specific pieces of code, try them out right here, and also eventually we'll talk about how to set up your developer environment on your computer and start incorporating all of these different snippets of scripts. After I do it, I can just copy it, okay? Copy to my clipboard and go to my RStudio or my Visual Studio to run it in R or Python. Okay, so importantly, here, the central place for this program is your OmicsLogic Data Science area, where you will see the sessions, you will see the courses, and also please explore the forum. On the forum, you will start seeing additional tutorials being posted about how to start with R and Python on your computer, how to leverage the different resources that we have. So you can see that Mohit Mazumdar here posted some tutorials on getting started with R and Python. So please review some of this material, because in our next sessions, we're going to be doing this together quite a lot. Okay, let me leave you with this. This is an assignment for our next time that we meet. Okay, number one, complete your profile on edu.tbioinfo. Okay, so please complete your profile. What is a complete profile? Please use your full name. Please add an avatar or a picture of yourself. Links to your social media sites and your research media sites, ResearchGate, OrcID, things like that. Okay, explore server.tbioinfo and code OmicsLogic.com. We'll be relying on these platforms, and we'll talk a little bit more about what these data science platforms are designed to do. They are designed to do analysis. Okay, yes, any code editor is going to be useful. We will first do everything together. We can actually track all of the activity on the code OmicsLogic.com, helping address any technical issues that you have. Okay, for those of you that don't have any technical issues, great. You don't need that. You can use it directly on your computer. What we will work in the sessions are the code OmicsLogic.com, RStudio, and Visual Studio for Python. Okay, where to find those? So you can see here, install R and RStudio and or Python and VS Visual Studio Code on your computer. Okay, so these resources are posted on the forum. Okay, so for those of you that are going to continue with us in our next sessions, please make sure that all of this is ready for our next session. Now, let me briefly tell you what you will learn and what is the schedule, so everyone is familiar with the schedule. Today, we talked briefly about data types and properties. It was a very brief introduction. I hope you found it useful. We will then continue to talk about different types of big data and what are some of the challenges and opportunities, where to find this data, how to load this data, and what can we actually gain from analyzing the different data sets. We will then talk about the independent steps of cleaning, loading, and processing the data. We'll run some pipelines. We'll give you an example. That example will come from Genomics 1, Transcriptomics 1, Metagenomics 1. You'll see how similar analysis approaches will produce different types of results. Once you have the data loaded, we'll talk about exploratory data analysis. So we'll talk about how to explore what's in our data and make some statistical conclusions about what we have. And we'll finalize the first week of the program with handling large and complex data. So then we'll talk about introduction to machine learning and AI, unsupervised machine learning, supervised machine learning, talk about model accuracy and validation, and how to go from learning about this to actual production. How do you become proficient and professional with what you do? Finally, I want to mention that many of you might come from an area of clinical research, biological research, and other areas of life sciences. We have many participants that have successfully completed these programs and have gained valuable insights about what they can do with their own data. How can they make their research more impactful? All of our participants, and we have several hundred that are already a part of the different social media groups. Beepsa, thank you for posting those links. We have them on Facebook, we have them on LinkedIn. Please join those groups. We will interact with you there. We will also help facilitate any connections that you might want with industry or research professionals. We also have an expanded research fellowship program that you're welcome to explore. These are designed to help those that are getting started with bioinformatics to apply bioinformatics to different types of projects. Again, anyone who's interested to understand what are the differences between this program and other programs on data science and bioinformatics, please explore on our blog. We have a few reviews by people that have done both. So you'll see what they think the differences are, and you'll see what people might be gaining from these programs. Again, thank you all for joining today. I'm going to open it up for questions. If you have any questions about registration, Beepsa is happy to answer those. You can reach her at marketing@pi.bio. If you have any questions about what we covered today, please put them in the chat for me or post them on the forum if you're already a part of the program. If you're interested to understand what kinds of projects we're going to be going through and using in this program, we have Dr. Mohit Mazumdar and Dr. Harpreet Kaur. They both have PhDs in bioinformatics in different domains, in chemoinformatics and in cancer genomics. So you're welcome to ask any questions for those as well. Do we have an internship program? Yes, our research fellowship program could be used for an internship as well. If you're a student and you have a requirement for an internship, please fill out the fellowship request and you will find the different options there. All right, well, thank you again for joining today. I have another session coming up, and thank you for joining and for all of your questions. I was happy to learn that so many of you are interested in data science, in machine learning and data mining. And please do email us and reach out to us. Unfortunately, this session was only scheduled for an hour and a half, but we will be excited to see you again. Our next session is coming up. So again, our next session is going to be on October 13th. I believe that is incorrect. It's going to be on October 14th. So please do join us. We'll be looking forward to seeing you, and have a great rest of your day.